Hello, and welcome to the Gene Regulation Room. Our exhibit is going to mainly focus on the operon model of gene regulation. To fully explain this, we'll begin with a prokaryotic cell, as you see in the bacteria here. Almost all of the genes in the bacteria is going to be found on the bacterial chromosome ring here. Some of it will be found on the smaller plasmids, but most of it will be on this bacterial chromosome in this big ring. So one large circular chromosome, as you can see there, a little bit on the plasmid, which is smaller loops of DNA. Now, operons are groups of genes that are similar and can be activated or deactivated. And they'll be found on the chromosome itself, some on the plasmid, most of them on chromosome, like right there and right there. And a little bit on the plasmid right there, as you can see in the little pink part. Now, the operon itself is located on the strand of DNA. And the first piece of the operon is the promoter region, which is right there, normally on the beginning of the operon model. And that is where the RNA polymerase will begin the transcription process by attaching onto that promoter region. So next, after the promoter, is going to be the operator region, which is right next to, which most of the time is right next to the promoter region. And now the operator is where the genes are either activated or deactivated by the, it can either be, we'll explain the rest of that later. And then the genes can code for functions in the cell or create proteins, whatever is needed as genes do, as you can see right there. So a good example of all this is going to be the lactose model, the lactose digestion operon. So lactose is a complicated sugar that has two parts here, and the bacteria needs to break that down into two different, more simple sugars to be able to use it to create ATP energy and use that in different various workings of the cell. And lactase is going to be the enzymes going to break down lactose. Galactase and glucose are going to be also the same thing there. So the lac operon by default is set in the off position. That means that the regulatory gene is still creating the repressor protein, which is this, that is the repressor protein right there. The regulatory gene is coding the mRNA right there to get onto that protein, which is going to latch on to the operator region. And when that happens, the RNA polymerase can't attach to the promoter region and therefore is unable to code for the rest of those lactose genes. So whenever there's no lactose, that's fine because the lactase is unneeded. However, when there is lactose found in the area, they will need those lactase genes. So what's going to happen is the exact same thing, except this time, as you can see, the lactose comes in and attaches to the proteins right there. So the proteins are unable to latch on to the operator system, and therefore the RNA polymerase can go through and code for the genes as is needed and create the enzymes here that are going to stop the lactose, and they're going to break them down and use them for the energy. And then after that happens, there'll be no lactose left, and therefore nothing to stop the regulatory protein from latching on to the system. So the lac operon will therefore go back to being off, and the cycle continues like that whenever the lactose is needed. So that right there is the operon model of prokaryotic gene regulation. On the other hand, we have a eukaryotic example that's going to be seen in this next slide right here. So gene expression in eukaryotes is basically much simpler than the operon model. Most of it is dealt with by these activator proteins that regulate how quickly the RNA polymerase, or how efficiently, maybe not quickly, can go through and transcribe these RNAs. And that's the biggest issue with gene expression in eukaryotes, are those activator proteins. However, it can also be managed by how the exons are cut in from the introns to create these different things. So as you can see, the two same genes here can be cut in two different ways to create two separate end products. The calcitonin, mature RNA for calcitonin, mature RNA for calcitonin gene-related proteins. However, gene expression can also be regulated by the efficiency of the RNA to go through the transport channels in the nuclear membrane. The mRNA must be allowed through by the channel itself. So whether or not that goes quickly or more slowly can be changed by the channel. And lastly, different amino acids and proteins are needed for the translation process. And whether or not these proteins or amino acids are there in abundance or sparingly can also affect the rate of the gene expression itself.